Okay, so I'm going to attempt to teach you about how adaptive immunity works. So this huge cell here is the macrophage, which is also a antigen presenting cell. And inside the macrophage is a lysosome. And you can see that there's a receptor here and also here. And these receptors are called MHC2, major histocompatibility complex 2. So macrophages have the type 2. You're going to see why that's important. So macrophages are part of the innate immune response in that they can eat anything that's foreign. So over here we have 2 evil pathogens and some nondescript pathogens. So one way that it can do that it can kill pathogens specifically is they can just take anything that's foreign, and what they're going to do is they're going to engulf it with phagocytosis. Oops. Hey, you. And please. Okay. And then that, of course, goes to the lysosome, and then it'll get digested. But that's not going to help us in the long run, because what we really want is to make antibodies, because antibodies make you well. So in order to make antibodies, we have to kind of have this macrophage signal the other lymphocytes. So what I'm going to do for that is I'm going to show you, this is going to be our pathogen here, this evil guy here. And when he gets engulfed through phagocytosis, what we're going to do is fuse him with the lysosome. And when he gets digested, I'm going to pull him apart into little pieces. And part of what gets digested, so this could just be like a protein on the outside of the virus or whatever, it's going to actually bind to this MHC2. And then, whoop, you didn't see that. And then when this vesicle fuses with the cell membrane, it's going to put, this is all just trash, it's going to put a piece of the pathogen on the outside of the macrophage attached to the MHC2. So this is going to be the signal to the T lymphocytes. Okay, so I'm going to try to move this around so that we can see how the T lymphocyte interacts with this. Okay, so this is dead pathogen. Bye-bye pathogen. Okay. So that's why this is called an antigen presenting cell, because it's literally presenting an antigen. So an antigen is just a piece of the pathogen, and it's on the outside of the cell. So here comes the helper T. Okay, so the helper T cell, this isn't any old helper T cell. This is a helper T cell that just happens to have the correct corresponding binding site to that antigen. So what the T cell is going to do is it's got a little thing called a CD4, which recognizes the MHC2 and says, okay, you're a macrophage. And then this antigen binding site binds to the antigen like this, and they connect. And so then the T cell says, hey, that's what I was born to recognize. That's it. That's the thing. So then what happens is the macrophage produces a protein called interleukin-1, which is a cytokine. And that activates the T cell to produce interleukin-2, which is, we're just going to make these pink things. And interleukin-2 causes the T cell to clone itself. So it's going to go and make 1 million clones of the same T cell that's going to have the same antigen binding site. So imagine 1 million of these now, ready to go. Okay, so the helper T cell is really important to communicate between the macrophage and the B cell. So here comes a B cell. I'm going to scoot you back, macrophage. Okay, so notice the B cell also has a little lysosome inside. This is the B cell. The B cell is also an antigen presenting cell. So he can find the same pathogen in the same way as the macrophage did. He's not big enough for me to show the whole thing, but can you imagine that he got eaten, engulfed, eaten in the same exact way? And on its MHC2s, it also has a little head. So then this will become an antigen-presenting cell, do you see? With the antigen on the outside. Okay. Too many things going on here. Okay, so now the helper T. Stop that. Stop that. Magnetic marble issues here. Hold on. Okay. So the helper T is then going to come and connect with the B cell. So here, this is an activated helper T, and here's a B cell with the antigen. And when this connects, this is going to cause the B cell to clone itself, but then more importantly, some of those clones will become plasma cells, and the plasma cells are going to produce antibodies. Okay? Antibodies make you well. If you can make antibodies, you're going to get well. So it's a matter for any kind of infection. It's, can you activate your B cells before you die? Because once you produce antibodies, you'll get well. So I'm going to show you how an antibody works and how it can be really effective to make you well. So an antibody is a protein, and there's two of these short proteins, and then there's two of these longer proteins down here. Right here is the place that will bind the antigen. So every antibody is going to be specific to the antigen that is here presented. So this antibody will only bind to that antigen. And so every single B cell will have MHC2 to bind to a certain antigen, which will also connect with the T cell. So these are all very specific. So the acquired immune response is very specific. It's not just eat anything foreign. It's this dude is what we're going to kill. So these antibodies are designed to fit that guy. So what I'm going to show you now is how an antibody works. But I'm not going to do it with the little man heads because I don't have enough of them. So I'm going to do it with these little puffball things. So I'm going to push all this aside. Just show you how antibodies work. Okay, so I have a bunch of antibodies. Remember, antibodies are proteins. So when the B cells become plasma cells, they are full of ER and they just produce so much of this protein. All the same antibody. So let's say this is our pathogen. These little yellow, they look a lot like viruses actually. So if this is our pathogen, the antibodies are designed to bind to the antigens on the pathogen, like that. Okay, so one thing that they can do, one way that antibodies can work, is if they just surround the virus, like let's say this is a virus, and they bind to the little proteins on the outside of the virus, it will prevent the virus from being able to bind to the receptors on your cells. So this is called neutralization, when you have antibodies just basically blocking all of the keys, so to speak, so that they can't fit into the locks on the cells. So they're binding to all the glycoproteins on the outside of the virus. That's neutralization. Another way that antibodies can work is called agglutination. And because antibodies have two little hands, they can kind of clump a whole bunch of pathogens sort of all together. And this is called agglutination. So now we have a whole bunch of these viruses all kind of held onto each other. And so now if a macrophage wants to eat them, they're not eating one at a time. They can eat large clusters at a time. And of course this could be three-dimensional, so you can actually get quite a lot of pathogens connected together with the antibodies. And then the last way that antibodies can work is they can bind to an infected cell, because the infected cells will present antigen on their MHC1s, And that's a signal that that cell needs to die. So when a cell is infected by virus, it'll present their antigen on their MHC1s. Their MHC1s are the normal signature proteins that all of the cells have. So when the antibodies bind to that, so let me see, if this were a normal cell here, and so let's say this is a normal body cell and this is the MHC1, and it has an antigen bound to the MHC1, this cell is basically signaling to the immune system, I'm infected with virus and you need to kill me. So the antibody will bind to the antigen, and that will signal the killer T cells to come and kill this cell. So hopefully that will help you to understand. I'm going to give you a bunch of other like professionally made videos that will help you too.